Hello class, today's topic is cell division in DNA. This is part 5. We're looking at a few of the same learning targets we've been looking at, plus an additional one on DNA sequences affecting organisms in different ways, which we'll call mutations. The other two are basically translation is a process we've yet to talk about, and again, how DNA is a molecule of heredity and makes proteins. So we'll continue to look at those as well. The process of translation is going to be something that is probably one of the most complicated parts because we're ultimately creating the protein. This is the end game here. This is what we've been talking about trying to make throughout this whole process. So in the last process of transcription, transcription made this mRNA molecule, which has the code for creating a protein. So now we're going to go ahead and do that and make our protein from this strand. So basically what we're doing is translating the mRNA message into that protein chain, which is called polypeptides or proteins. You can use those interchangeably. That's all right for this class. Amino acids are the things that are going to be the building blocks of our proteins. So we'll call them the language of proteins. There's the 20 amino acids that you'll start to be exposed to. We won't memorize them or anything. We'll use a codon wheel, and you'll see what that looks like. Basically, these amino acids are kind of strung together to make proteins, just like letters are strung together to make words, and nucleotides are strung together to code for those amino acids. So it's all kind of the same idea that there's some code, and that code that's put forth is going to allow us to have specific series of amino acids put together to make specific proteins. Codons are 3-letter nucleotide sequences of the mRNA that code for a specific amino acid. An anticodon is a set of 3 nucleotides on a tRNA. So remember to keep them different here. Codons are always on the mRNA strand, and anticodons are on the tRNA strand. These two things will match up, and you'll see that in an image in a bit. But basically, anytime we're talking about anticodons, they're always on the tRNA. The easy way to remember is that codons are on the long strand. This is a long strand. And tRNAs are kind of free-floating. So you'll see that in animation. All right. Example codon, a GGG codon that's on the mRNA is going to pair with a CCC anticodon on the tRNA. So with all that codon-anticodon talk, we'll start to see an animation down here that hopefully makes a little more sense. Step 1 of translation begins at the start codon, which is an AUG. The start codon is going to be, we'll abbreviate as this. MET, this is an amino acid called methionine. Methionine is the amino acid that proteins start with. That tells the whole process where to begin. Remember, we have this long mRNA strand. There's a lot of information on it. Where we begin is an AUG or a start codon. All right, this probably should have been read too, start codon. Step 2, the tRNA contains an anticodon that binds to the mRNA's codon, and it brings with it an amino acid, whichever kind it's supposed to. So in this animation here, we'll take a peek at it and identify all the parts and things that are here. First of all, we have rRNA. This is an important molecule. This is this big purple blob here. It's got a large part and a small part, which we don't need to get into, but the idea that this rRNA molecule is kind of clamping on to everything to help this process unfold and give it a place for these amino acids to come in at. This thing down here is messenger RNA, mRNA. That has the code. That's the thing we made in transcription. We got this one single-stranded RNA molecule. And then these guys flying in here, they kind of draw them here to the chemical structure to actually look kind of like a T, but these transfer RNAs are flying away, and that's what's coming in. So we have tRNA here. All right, that's those molecules. Basically, all these things play a part in making the protein. So what we're talking about in step 1 is that we have the start codon, this AUG, which you can see right there, started the process. and then these tRNA molecules will come in. The tRNA molecule has an anticodon, and the RNA molecule has a codon. So you see them match up right there, codon and anticodon. When they match up, they bring together these little amino acids, and the amino acids are being strung together to make a protein. Once enough of them stick together, we'll reach a stop codon and the protein will break off. That's our polypeptide chain. So there's a lot going on in that animation, but a really important process to visualize because it sounds really complex. When you see it, it doesn't look so bad. Step 3 and 4. In step 3 of translation, the amino acids are going to bond together, and then the whole thing stops, like I said, when it reaches a stop codon, and the newly formed polypeptide is then released out into the cell. So stop codon is important, and I'll show you how we identify all those, and then the polypeptide or protein is then released out of there. Determining protein sequence. Basically, because of the mRNA strand is in order, we start at a start codon and we move through and we continue to bring amino acids. And here's a wheel of our amino acids we were talking about. These can be formed in different orders, in different ways. They fold up on each other in special patterns to make very specific proteins, which are important in functioning of any living thing. The codon wheel, how do we determine what amino acid comes in when? We have the mRNA code, we understand it, so there's a nice little wheel for this, and I'll give you an example. If we had an mRNA that was, let's say, GAG, what would that be? We had GAG. Well, GAG, how do we figure that out? We start in the middle and move through. So we have G, and then I move to A in the next ring, then I want to find a G in the last. So this process of GAG equals glutamic acid. So you could go through that whole process for any codon to figure out what its amino acid might be. What I did want to highlight to you guys was the stop codons are kind of dispersed out. So you see different stop codons. UAA is a stop codon, UAG is a stop codon, UGA is a stop codon. So you see those scattered throughout. The start codon, AUG, we talked about that one. Go A to U to G is this methionine, which is our start, which kicks off the whole process of building our protein, bringing in that methionine amino acid. The central dogma of biology is really this, and really wanted to talk about what this is. The central dogma is that DNA is this foundational molecule contains information to make proteins. So to put it all into play here, here's the process of all the notes we've been talking about since DNA structure. DNA needs to replicate so that our cells can have information that they need when they divide and grow. So replication is important. What is that process? DNA to DNA. We just continue to copy DNA and move it into the right place. When the time comes, then we take our DNA and we create RNA from it. So this process is our next process, which is transcription, which is right here, right? So this transcription process. And then finally, we take that RNA molecule and we translate it into proteins. So we see that whole process unfold, and that's our central dogma of biology, is that DNA goes through these steps to make proteins. The final thing we need to talk about is mutations. Mutations are a change in the organism's DNA. Basically, DNA can be altered through a number of ways, but when DNA is altered, it's typically not a good thing. So we'll talk about the effects down there in a sec. This can happen during the replication process, and it often can affect a single gene. If your mutation is not fixed, it may permanently damage an organism's DNA. So what are some things that could happen? Basically, we could have a genetic disease if there is something wrong in the code of DNA. And we'll look into some of those. Could result in cancer. So in the image over here, we see this is ultraviolet light or UV light. Can affect the bonds in the DNA molecule and actually can make what we'd call a thymine dimer. Basically, this yellow one is a thymine. So you have a thymine and a thymine on the same side stick together. So this UV light can cause changes in the DNA structure that way. It could also lead to new genetic traits, so just something that's a little different. This is kind of a small, a part of how natural selection works, which we haven't talked about yet. Or it could lead to no harm at all. It could just be a harmless mutation that doesn't affect a single gene that really matters, and so nothing really can come of it.